Hello, my name is Marshah Menos. My mentors are Dr. Zhao Ping and Dr. Frank White. I attend the University of Florida, and I am a Ronald E. McNair Scholar. The title of my project is The Function of Type 3 TAL Effectors in Bacterial Canker of Mango. So what exactly is causing bacterial canker in mangoes? There's a bacteria known as Xanthomonas citri PV mangifera indica, which is a form of gram-negative bacteria known to be the causal agent of bacterial cankers. Some of its common symptoms include black leaf lesions, chlorotic halos, and in the most severe cases, premature fruit drop. The photo you see on your right is an example of Xanthomonas citri on mangoes. But what exactly are TAL effectors? They're actually a form of protein that's created within bacteria. So when the bacteria goes into affect the host cell, it's going to be releasing these proteins into the host nucleus. Once the protein is in the host nucleus, it's going to be targeting the host gene promoter in a sequence-specific manner. From there, it's going to result in gene expression. So now the host gene is now going to be promoting the disease and causing the bacteria to replicate and spread. So my hypothesis is as follows. TAL effectors of Xanthomonas citri PV mangifera indica function in bacterial canker of mangoes by targeting specific mango genes, resulting in ectopic expression, and the protein product of the expressed host gene facilitates the infection of mango leaves by the bacterium. So that leads me to have two predictions. One, TAL effector gene mutants of Xanthomonas citri will have phenotypic changes in the disease symptoms, and two, specific TAL effectors of Xanthomonas citri will cause dramatic expression changes in specific mango genes upon infection by bacteria compared to bacteria with mutations in the respective TAL effector gene. So for the procedure, my objectives are to generate TAL effector gene mutations in Xanthomonas citri by targeted mutagenesis and examine mutants for effects of symptoms and host gene expression. So the first step is going to be examination. The genome of Xanthomonas citri was already sequenced in the Dr. White lab, and the TAL effector genes were already identified. So I'm going to be selecting a certain portion of the Xanthomonas citri gene for PCR implication. So I'm going to be preparing genomic DNA from the Xanthomonas citri strand for the PCR process. Step 2, PCR. I'll be utilizing a polymerase chain reactor, PCR, to amplify a subfragment of a TAL effector gene found in my Xanthomonas citri strand. This will be done using forward and reverse primers. So the amplified fragment will now be cloned into a suicide vector known as p-topokinase. However, p-topokinase cannot replicate a laboratory strand of Xanthomonas citri. The only way for the p-topokinase and TAL effector genes to survive, or in this case replicate, will be through homologous recombination between the subfragments in p-topokinase and the TAL effector genes in Xanthomonas citri. This will give us several mutant strands of Xanthomonas citri. Step 3, electroporation. So p-topokinase TAL effector gene subfragment clones will be introduced into Xanthomonas citri via electroporation. The subfragments will be prepared. Xanthomonas citri cells that are mixed with the vector DNA are going to be shocked by an electric current. During this process, the cell membrane becomes permeable, allowing the vector DNA to enter and homologous recombination to occur between the p-topokinase and TAL effector. Selection for resistance to antibiotic kanamycin will show which Xanthomonas citri cells have been able to successfully complete homologous recombination. Step 4, PCR Part 2. To ensure the kanamycin-resistant strains of Xanthomonas citri have gene mutations, we're going to perform PCR again with the TAL effector genes. We will be using different primers and as well as a different vector DNA primer. After verification by PCR, we then have obtained multiple mutant strains, which we can now use to test on mango leaves. So in conclusion, this experiment will help us in understanding whether type 3 TAL effectors are actually being utilized when bacterial canker is infecting mangoes. With the success of this experiment, we could hopefully proceed further in determining which specific host genes are targeted when TAL effectors are attacking. Further experiments will be designed to understand why specific host genes enhance the disease process. So this is my Works Cited page, and this is my Works Cited page continued. And I would like to give a huge thanks to my mentors, Dr. Zalping and Dr. Frank White. They were there to help me throughout this project this entire summer, and I don't know how I could have done this without them. And a huge shout out to the McNair program for allowing me to do research this summer.